Ladies and gentlemen, thank you very much indeed for your patience. And as I say, good evening, everyone, and from wherever you are, and welcome to the Ulster Beekeepers Winter Webinars. My name is John Hill. I'm the chairman of Ulster Beekeepers Association. And as I say, I'd like to welcome you all. This is a series of webinars which we're putting on in place of a conference that we normally run about this time of the year. This is the third webinar in the series of 7, and the rest will be continuing every Wednesday until the middle of February. This evening, the webinar will be by Professor Robert Paxton from Halle University in Germany, and he will be speaking really on bee viruses, past, present, and future. Our sponsor for this particular webinar this evening is Simon the Beekeeper. Simon runs a bee equipment company in Nuneaton in England, and he actually became interested in beekeeping after a heart transplant back in 2005. So we're delighted, sir, that he's still with us, hale and hearty. He made a natural progression from being a keen gardener with an allotment to be a novice beekeeper with a motivation for reintroducing honeybees in his own village. And his admiration and respect was kept growing for the species which play such a vital part in the world's ecology. So we're delighted really that Simon the Beekeeper is our sponsor this evening, and we're very grateful for his help in helping us to stage this webinar. Now, as I've said before, there is a chat line on your Zoom screen, which we'd like you to use to tell us where you're from. And then there's a Q&A button in which you're able to ask questions. We will be having a question and answer session at the end of the webinar. And certainly if you wish to ask a question, please use the Q&A button. Now, we will have a period of time at the end in which Robert will, in fact, answer questions. But in fact, if there's more than that, he has offered that he is quite willing to answer questions by email, and his email will be displayed on one of the slides that is about to come up. So if you miss your chance of having an answer question at the end of the Q&A session, then certainly please avail of his offer to answer your questions by email. Now, I would really say the other thing really is that this is a free webinar. Ulster Beekeepers are putting on this series free of charge, and we're very grateful to our sponsors for helping us to do that. However, if you or your beekeeping organization wish to make a donation towards our expenses in running that, we would be very grateful. And certainly if you wish to do so, if you could please contact the Ulster Beekeeper Treasurer, and his email address is ubkatreasurer@gmail.com. So that's ubkatreasurer@gmail.com. Again, as I say, it'd be much appreciated. So, ladies and gentlemen, we're going to move straight on, and if I could, in fact, introduce our speaker, Professor Robert Paxton. Now, Robert, in fact, is a graduate of Sussex University, where he undertook a PhD on sex ratios in solitary wasps in 1985. And then Robert subsequently learned all about bees and beekeeping under Professor Robert Pickard and John Free of Cardiff University in the late 1980s and early 90s. He moved to Uppsala University in Sweden in 1993, and then to the University of Tübingen in Germany in 1996, where he researched the genetics and social evolution in bees before moving to Queen's University in Belfast in 2003, where he expanded his research to include pollination by bees and conservation genetics. In 2010, he moved back to Germany to the University of Halle to take up the chair in zoology, where he has re-established his group working on bee biology, including their parasites. His main research areas are social evolution, host-parasite relations, pollination, conservation genetics, and bees remain at the core of his studies, with a recent focus on viruses, particularly deformed wing virus and its interactions with honeybees and wild bee species. He has for long been interested in the honeybee, which features strongly in his research program. So, ladies and gentlemen, I welcome Professor Paxton, and Robert, I'll hand over to you.
Thanks very much, John. Thank you very much for the introduction. I shall share slides, and I hope that you can see and hear me, because if you can't, I can't respond. So please wave if there's some problem.
Right.
Okay?
Yes,
Yeah, good.
That's fine.
Good. Yeah, great. So thank you very much, John, for the introduction. And I'm going to talk about bee viruses, some research that we've been doing, looking at the past, present, and say something about the future, at least in terms of control. Certainly, I'll be saying something about pathogen spillover. So if I got this right, let's see. Yes, so here we go. There I am in the red circle. That's Halle. That's where I'm sitting at the moment, just before bedtime. You're mid-evening, I think, for most of you anyway, if you're in UK or Ireland at the moment. And in Halle, where I work, where I'm at university, we have a couple of alumni. One of the city, Friedrich Händel, famous Baroque composer who was born and brought up. There's the house on the right where he was brought up. And maybe I can, which is in the city centre. And I can use the laser pointer maybe. So there we go, his house, and there is his bust in the city centre. And also Martin Luther, who was one of the first professors of the University of Wittenberg that was taken over by Halle to become University of Halle-Wittenberg after the Napoleonic Wars. So that's Martin Luther, another alumnus of the university. But now I'm going to preface this talk by introducing SARS, or mentioning SARS. SARS-CoV-2. It's a virus that we're all acutely aware of, and I think it helps to reinforce this idea that viruses are everywhere and they have major impacts upon animal populations, upon our own health and well-being. And we're also acutely aware that there are different variants of SARS-CoV-2, the virus that causes COVID disease. Omicron is now present everywhere. I just downloaded this afternoon these images showing in the UK how numbers of corona cases, positive cases, have skyrocketed up to almost 200,000, and that's 16th of January 2022. Massively. In Germany, it's not been so different. We had a Delta peak, a peak with the Delta variant towards the over Christmas time, and then it subsided over Christmas, and now we're up to over 100,000. That means yesterday we had, I think it was 120,000 new cases of SARS-CoV-2 reported in Germany, the highest it has ever been. And a large proportion of those are the new Omicron variant. Okay, so I started off with SARS-CoV-2, and we're all acutely aware of it. Now, what about our bees? Well, I've used this image quite a bit to say that, which is, I think it's a nice image that shows what's happened, at least in the US in 2006, 2007, with massive colony losses, lots of dead bees. I'm sure many of you have witnessed this in your own colonies, bees dead at the bottom of the hive, particularly coming out of winter, when apparently there are still stores available. And we've certainly had this as well in Halle, despite our greatest efforts to control the varroa mite. And it's why the varroa mite is a major cause of this collapse of colonies or massive loss and die-off of colonies. And not only the varroa mite, but also probably the actual cause is the viruses that are transmitted by the varroa mite, like deformed wing virus, which causes deformed wings, as you can see here in this adult honeybee, when it's parasitized as a pupa. But there are other viruses, of course, that are also associated with honeybees and that are transmitted by varroa mites as well. Okay, why do I focus on deformed wing virus more recently? Well, it's because it seems as though it's the most prevalent virus that causes colony loss. Let me show you some data now that I and colleagues have collected that try to identify what are the viruses that are associated with varroa mite ectoparasitism of honeybees. And this is part of a former EU project in which a large number of colonies, this is over 600 colonies, were sampled. Sampling means taking 30 bees from the colony in 4 locations around the world that had in one part of that location in red varroa mites in their honeybee colonies, and in another part of that location, in this case in Newfoundland in Canada, green at the time in sampling 2010-2014, honeybees that were without varroa mites. The same in the UK. And I must say the UK data come from Northern Ireland, actually, that are with varroa mites. And the green are islands, and in particular Colonsay, and thanks to Andrew Abrahams for providing those bees, for allowing us to sample these colonies that were without varroa mites. You can see in Scandinavia and in New Zealand at the time, Southern Ireland devoid of varroa mites. So by doing this sampling across multiple locations, we hope to control for any regional effects that there might be, and just be able to concentrate on what virus is associated with varroa mites versus not associated with varroa mites that are parasitizing honeybees. Better said, what viruses are associated with honeybees when they are with varroa mites or without. I have to thank a lot of people for the data that provided this. It's in preparation now for publication by Melissa Oddy. But the data I can show you, because these have already been analyzed. Here are pie charts showing the proportion of colonies that were with a virus or without a virus. The viruses are listed here at the top. There are 16 of them that we screened. So black queen cell virus here, for example, the commonest virus, and the top row of circles shows the proportion of colonies from those 4 locations without varroa mites, that were devoid of varroa mites. Lucky them. And below are the prevalences of the viruses, of those 16 viruses, where varroa is present in those 4 locations. And what you can see is, actually, interestingly, there's a lot of virus in honeybees. They're often parasitized by virus. The commonest virus we found was black queen cell virus. Deformed wing virus comes in 2 genotypes, which I'll mention in a minute, was not the commonest, but certainly increased in prevalence with varroa mites. So these little stars at the bottom show which viruses increased in prevalence with varroa mites. And you can see black queen cell virus as well became more prevalent in colonies. There were more colonies with black queen cell virus in them than with varroa mites than without. So there's quite a number of viruses that are more common in honeybees with varroa mites. So varroa mites seem to cause dirtier bees, unfortunately. Now, what about the quantity of virus in those mites? Because we've used a technique called qPCR, quantitative PCR. It's the same method that's used for detecting corona and its variants. And we've used that for detecting virus, quantifying the virus in the bees. You can see here the figure, those colonies that didn't have varroa mites, generally they have about, I can see here, it's about 1 million virus particles in them, or genome equivalents, strictly speaking. Honeybees with varroa mites have about 10 million viruses in them as a median value. So they are dirtier. They have a higher parasite load. And if we look across those 16 viruses, there's one virus that is more prevalent, that is at a higher titer with varroa mites than without. One that's statistically significantly higher, and that's DWV genotype A. Genotype B looks as though it's different, but looks as though there's more B, but there are very few sites. So black queen cell virus is marginal significance. So if we summarize those data, what we see is that 2 viruses black queen cell virus possibly, deformed wing virus A definitely, is both more prevalent, it's found more often in colonies, there's more colonies that have it, and where it's found, it is at higher titer. And it points the finger at deformed wing virus A being the principal virus that is associated with varroa mites, or was when we undertook that study. And this is this result really just reinforces what Steve Martin has shown on the islands of Hawaii in 2012, that when varroa entered the islands of Hawaii, which is in red, those islands in red, then the viral titers in those islands with varroa mites shot up in comparison to islands that hadn't been invaded by varroa mites. So, and it's deformed wing virus. This is the viral titer of deformed wing virus, whereas other viruses didn't change that much. So there's strong evidence that deformed wing virus is associated with varroa mites, the major virus that's associated with them. Now, which virus is associated with mortality in honeybees? Well, that's a bit of a difficult question to answer because we don't often do experiments at the level of the colony. But I'm going to recount some data that we generated when I first moved to Halle. We went to visit colonies in the southwest of Germany, Black Forest, and we sampled 28 colonies from 2 beekeepers in 2 separate apiaries. We sampled those colonies in, let's say, September, October time at the end of brood rearing. We collected adult bees, 30 bees, and we screened them for virus. And then we collected another 30 bees from 28 colonies. All of them survived, some of them just about survived, but all of them at least had some bees in them in springtime. And we collected another 28 bees, another 30 odd bees from each of those 28 colonies and screened them for virus. And this is the pathogen, and not only viruses, but other pathogens as well, and nosema. And this is the data that we found. So in autumn time, on the left, what you can see is the prevalence of virus in colonies in autumn and in spring. And this is prevalence of deformed wing virus B, which otherwise is called VDV1, used to be called VDV1. So what you can see is all colonies, more or less, had the virus in autumn time. In springtime, they didn't have it anymore. So presumably, the autumn bees that were infected, that caused this prevalence, were all dead, and most of the bees that survived were the ones that didn't have the virus inside them. Let's skip the middle thing. Most importantly, on the right hand side that I'm showing here now, we could record the change in the number of bees over winter. So we measured the size, we used the so-called Lieberfeld method to measure how big the colony was, how many honeybees it had in autumn time, going into winter, and then how many in springtime when it was coming out of winter. And some colonies only lost 10% of their bees. Some colonies lost 70% of their bees. This is how many bees were lost over winter. And this is the prevalence of the virus in, I think we collected in this case, 10 individual honeybees in autumn. And what you can see is there is a statistically significant relationship. The more bees that were infected in autumn time with deformed wing virus, genotype B, the more bees were lost from the colony over winter. And when we look, the other virus that we found in large numbers, amount, was black queen cell virus. We get sort of the same pattern in autumn time. More or less every colony has black queen cell virus. In springtime, hardly any of them have black queen cell virus. Conclusion, those bees that have black queen cell virus do not survive over winter, and those that go into winter without it do survive. But there's no correlation between black queen cell virus prevalence in those worker bees in autumn time in a colony and the percent of the colony that's been lost. Boom, no relationship. Only with only with deformed wing virus. When we look at the nosema, we found both Nosema apis and Nosema ceranae in both in the colonies. Again, no relationship with the proportion of bees infected with Nosema and colony loss. So the only pathogen that we found that was correlated with decline of a colony was deformed wing virus B. which again points the finger at deformed wing virus B. Now, I've talked about deformed wing virus and now deformed wing virus B. Like corona, deformed wing virus comes in multiple variants. This is the virus, at least this is what the virion looks like under a cryo-electron microscopy, electron density image of it. It's a very simple virus in that it's a single positive strand of RNA. So it's a positive single strand RNA virus. So is corona. It's only 10 kilobases long, 10,000 bases long in its RNA sequence. Corona is about 30,000, it's 3 times bigger than that. So there are some similarities, but it comes in 2 major variants that are widespread. Let me put it like that. Genotype A and genotype B, and these differ by about 17% in their sequence. So they're actually quite different. It's not like corona in that case. You know, corona variants, Omicron, Delta, they are quite similar. They're like, we're talking about 98-99% similarity. These things are quite divergent, and they're probably better termed genotypes A and B. So we were quite surprised actually in the Black Forest, because deformed wing virus A is supposed to be widespread, and it's the common variant that's been well described. And we wondered, why did we only find deformed wing virus genotype B in our colonies? Well, that may be revealed in a few minutes. So we saw that DWVB was killing bees, so we wondered, well, is it more virulent than A? Is that why it's and therefore more transmissible because it replicates faster? Why do we find only B, not A? So on the basis of that experiment that we saw, or rather that set of observations in the Black Forest, we set about testing whether one virus, the genotype B, was more virulent than genotype A, that apparently had been found everywhere and was widespread. And the best way to do that is with an experiment. You know, if you've got an idea, hypothesis, run an experiment. And I'd like to thank here Dino, Vincent, and the 3 people in the lab who ran this experiment. We extracted DWVA from an individual honeybee and used it as our inoculum. We extracted DWVB from a different one of our Black Forest honeybees and used it as an inoculum. And then we inoculated hundreds of worker bees, either with A or with B, or with a crushed up bee that didn't have the virus in it, or with both, and we don't have to worry so much about that. And then we injected them, put them in cages. Here we've got the experiment in which we put 21 honeybees per cage. All honeybees in a cage were infected with one or other of the 2 genotypes of deformed wing virus. We had multiple cages. I think in this case we had 3 cages per treatment. And then we kept them in the laboratory and waited until they died. Cages, they've got a feeder, they've all been injected with one or other of the genotypes, so it's all highly controlled experiments, but it allows us then to say objectively, do we have difference in virulence, at least at the level of the individual bee in these viruses? And these are the results, which I'll take you through, and I'm going to use these survival curves later as well. So let me explain. On the y-axis we have survival, going from 0, everything's dead, to 1, everything is alive. And here on the x-axis we have days since the start of injection, or days post-inoculation. At day 0, when we inoculate, every honeybee is obviously alive. If we inject bee juice into them, they follow this the mortality follows this black line, and by about day 40, they're all dead. And that's typical for honeybees that you put in a cage and feed with sugar syrup. They last in summertime about 40 days without you doing anything to them, just feeding them. DWVA in blue here kills them much faster. DWVA is a killer. Yeah, it kills bees. It more or less, so it's about a third of the average longevity of a control bee. DWVB kills them even faster. and it's only 4 days difference that it kills them faster than A. So B is also a killer, and it seems to be, from this experiment, a worse killer. It kills, it's more virulent than DWVA. Okay, the statistics, I'll spare you the formal statistical analysis of those data, the Cox proportional hazards model, but I shall go on to say we use the data from this experiment and to parameterize a mathematical model called Behave, which Juliet Osborne and her group have developed in Exeter, the University of Exeter, and we just put our data for the virulence of A and B into the model, and it models the dynamics of a honeybee colony with varroa mites and with virus. And what we found is, at least this is just theoretical, just a theoretical model, we find that A kills honeybees, a colony of honeybees, in 4 years, and B kills a colony of honeybees in 3 years. Now, that is just an abstract of a mathematical model, but it tells you that the difference in mortality between B and A, it's only 4 days on average, but it can probably make a significant impact on the speed with which a colony dies when it's parasitized by varroa mites that are transmitting DWVB. Now, that experiment was done on adult honeybees because we were interested in what happened to our colonies over winter when varroa mites overwinter with the adult honeybees. There's no brood, and they are feeding and moving from one honeybee to another and feeding on those honeybees. Of course, you will tell me varroa mites usually feed on, it's summertime, are feeding on the prepupae and the pupae, as you can see here in this diagram, and this is where they mainly feed and where they mainly reproduce. And once the pupa emerges, those adults that have survived or have bred within the on the pupa, then they're phoretic only for a few days or even just a day or two, and immediately then jump into another pre-pupa or another L5 late instar larva. True. So we said, okay, let's repeat our experiment comparing the virulence of DWVA and DWVB, this time on pupae. We used white-eyed pupae. This is much easier to do because we don't need to put them in cages. We then took white-eyed pupae and injected them with either A or B or control solution, or both. And then we quantify, we see what sort of mortality there is. We can also quantify how much virus is in them. And what we found, I guess a little bit to our surprise, and maybe I shouldn't be surprised, that there was no difference in the mortality of pupae injected with A or B. So here we've got proportion of dead pupae. And again, we've got several hundred pupae in this experiment. It's fairly easy to run these experiments. Control pupae, we lost about 10% of control pupae. You just inject a pupa with bee juice, roughly 10% of them die, 90% survive. So it's pretty good survival, but it's not 100% because of course, we're damaging the pupa a little bit. If we inject A into them, there's about 30% mortality. So there's about 20% higher. If we inject B into them, it's about the same mortality, no difference. And my guess is that, most of you are beekeepers. We injected white-eyed pupae, and probably the time that it takes for a white-eyed pupa to emerge, when we say it's successfully emerged, it hasn't died, is about 6-7 days. And that's probably not long enough for devolving virus to kill a pupa. And I guess that's why we don't see any difference in virulence. Whatever, there's no difference in virulence in the pupae over a period of 5 days. In adults, though, over a period of 15 days, there is a big difference. And, I think actually it probably, on reflection, it probably makes sense that the DWVB, though more virulent than A, and probably growing faster than A in a host, so it makes sense that it doesn't kill pupae too quickly. And that's because its transmission is dependent upon varroa mites successfully breeding and emerging in a pupating, an enclosing adult honeybee. there's probably some sort of transmission threshold here in the honeybee varroa mite system. So imagine we've got a varroa mite, enters an L5 larva. It then has 12 days in which it can start to feed. I think many of you know, most varroa mite females will, on a worker honeybee in temperate climate conditions, produce roughly 2 daughters and 1 son, so that when that bee emerges, there'll be mum, 2 daughter offspring, and a male which dies immediately, to transmit the virus further. DWVA, I suspect, has a virulence down here. If this is a virulence bar here, with low virulence here, I've got 2 low here, and 2 high virulence up here, there's probably some threshold here. If a virus is down here on this virulence threshold bar here, which I suspect DWVA, I'm going to suggest, is down here, when it infects in a varroa mite, when it's transmitted to the pupa survives and emerges, and DWVA can be transmitted further. A virus like acute bee paralysis virus does not survive in honeybee colonies, particularly when varroa mites are present. It's an absolute killer when transmitted by varroa mites, and within a few days the pupa is dead, and that's the end of the pupa, and that's the end of the varroa mites. That's the end of transmission of APPV, hence the threshold that is here. My suggestion is that DWVB is slightly more virulent, so it's slightly higher in virulence than A, certainly in adults, but not virulent until pupa, and therefore it is below this transmission virulence threshold. It survives very well and is transmitted readily by varroa mites. Okay, so B seems to be then, my suggestion is, and our experiments have shown quite clearly, and these are experiments that we've repeated now, I think 5 times independently, we find always B is more virulent than A. And I think I'll just say that Madeleine Beekman has suggested in a recent paper with her group working in Australia and the Netherlands, that they find the same as well, that B seems to be more virulent than A. It's not an experiment that has been designed to test that, but there seems to be independent confirmation that B seems to be more virulent than A. So if that's the case, and it's transmitting very readily, then maybe it's spreading. And that's the hypothesis that we've had, that DWB, in fact, when we published about the change in virulence of B, the higher virulence of B over A, we suggested at the time in our paper that probably B is going to spread. Now, where is B? First of all, let's cover varroa. We know varroa originated in South and East Asia and is being transported through to Europe and South and North America, across the world, by us humans. So it's originated from Southeast Asia. That's very clear. Deformed wing virus A, from sequences, from phylogenies generated from sequences of deformed wing virus, deformed virus seems to have originated in Europe. So Wilford et al. Have a very nice paper showing fairly convincingly that deforming virus genotype A arose in Europe, and probably varroa that was transported across to Europe picked up deforming whilst feeding on honeybees in Europe, and has subsequently, presumably by legal or illegal means, been through transport of honeybees and the varroa mites, has been spread around the world on European honeybees. So deforming virus A, most probably of European origin, and spread then around the rest of the world, everywhere except possibly Australia, where varroa mites, or at least varroa structure, is not yet found. What about deforming virus genotype B? So let me reaffirm. This is genotype A, worldwide, except Australia. What about genotype B? Well, it was first reported in the Netherlands by Ongus et al. Picked up in a colony published in 2004, but the colony that was the honeybees that were picked up and examined were actually collected in 2001 in the Netherlands. It was the next report of it was in Israel, 2008. So this is the first time in a country that deforming virus B has been reported. You can see then France and Germany later, then South Africa, 2010. We found it in Germany and the UK in 2011. East Africa, Georgia, we found it 2014, again 2016 in Europe. First recorded in Canada by us in 2011. In US, first recorded in 2012, mainland in Hawaii 2012. Latin America a little bit later it was first recorded, and China also a little bit later. So it seems as though deforming virus B is now being reported more widely. It seems as though it's spreading seemingly from Europe. And although one could argue that maybe it's lack of detection, lack of recording that's meant its lack of detection because people didn't look out for it before. If one scours through the GenBank database of sequences, one can find lots of DWVA sequences in next generation sequences, transcriptomes, but not DWVB, suggesting that genuinely B is something relatively new on the scene. Okay. What about its epidemiology then? We have just shown you where it's found, but what about its epidemiology? There we have more data, and the first data that really show this very clearly, change this is a change, epidemiology, change in the prevalence of the virus genotypes A and B. This comes from Eugene Ryabov. This is not my work, this is not my group's work, this is Eugene Ryabov in US Department of Agriculture. Showed in 2010, samples from 2010 honeybees in USA had mainly A. There were 2 records of B. By 2016, everywhere there was B, more or less, as well as A. Of course, A remains there. And I've used his data, just transformed them into this format to make it a little bit easier for you, because I'm going to show you now how the pattern, how the prevalence of deforming virus genotypes A and B have changed. What we see from Eugene Ryabov's data, so this is just his data reshown, is that in 2010, all colonies had A, very few had B. In 2016, still a lot of A, but B has increased in prevalence quite markedly. Now, we sampled honeybees in 2011 at 26 sites, collecting 30 adult honeybees at flowers. So these are ostensibly healthy bees, collected at these 26 sites that you see in green across GB and the Isle of Man, and including Colonsay. And we found DWVA, these are heat maps showing the prevalence of DWVA and the prevalence of DWVB inferred across GB. What you can see is a concentration. There seems to be a high prevalence of DWVA in around East Anglia, and a high prevalence of DWVB around East Anglia as well, and also in the southwest of England. So we've got data from 2011 sampling of bees collected in flowers. We went back to some of those same sites, 7 of those same sites in 2017, and collected from the same sites another 30 honeybees, or a number of honeybees foraging on flowers, healthy bees, and screened them for DWV A and B. And this is the 2017 data which are here. And what we find is that in 2011, at bees and flowers, A was more prevalent than B. Now 2017, or 2017 is a few years ago already, A was less prevalent than B. B was more prevalent than A. There seems to be a switchover. And these, incidentally, are almost identical results to that found published by Kevill on British honeybees in a recent publication. Now, we've been mathematically modeling the algebra and the differential equations, but given that B is if it is possibly more virulent, and possibly more transmissible because it grows to higher titers than A, then we would imagine if there is no interaction between A and B, we're going to get A, increasing in prevalence in a population. If this is time and this is prevalence of a genotype in a population of honeybees from 0 to all honeybee colonies infected. And we expect B also to increase in frequency, so that after time, we would expect at equilibrium, both A and B to be equally common, because both are highly transmissible through varroa mites, both are kill honeybees, admittedly, but they build up large numbers of virus within their hosts, which are then transmitted by varroa mites. So let me show you now, ask the question, does DWVB then reach an equilibrium prevalence with DWVA? And here, I did hear that Mark Schaefer was in the audience. If he's in the audience, Mark, I want to say thank you very much. Mark kindly looked at some honeybees that were sent to him in Germany, and Dr. Mark Schaefer is the person responsible in Germany for honeybee health and bee diseases, works in a state institute in northern Germany, which is specialized in viruses. And he kindly went back and looked at bees that had been sent to him from across Germany that have died, that have been collapsing or have collapsed and died. So he went back and analyzed data which he'd screened for DWVB, but not looked at the genotype. And what he found is for these colonies that were sent to him, or the bees that were sent to him in 2013, half of them had DWVA in them. All of them, more or less, had DWVB in them. I was flabbergasted. My goodness, that means DWB was already very widespread, I'll suggest, in Germany by 2013. Reflecting back, it is no wonder when we went to the Black Forest that we only found DWVB in our bees. You know, if it's so widespread, of course that's why we found in 2011 in the Black Forest only DWVB. In 2019 and 20, all colonies infected, all collapsing colonies were infected with DWVB. Only 10% of colonies were infected with DWVA. Obviously, those 10% also had DWVB in them. So it doesn't look as though this virus is reaching an equilibrium. It looks as though DWVA is being out-competed by DWVB. And these data, Mark, I should tell you, I am in the process of writing up for publication with you as co-author. So thank you very much, Mark, for the data. Now, last week, Francesco Nazzi, another honeybee researcher from Italy, kindly sent me some data from some of the bees that he has been studying. And he has been working, many of you may know him from his work. He's an experienced honeybee and varroa and virus and pesticide researcher doing some really important research. I asked him whether he'd screened for his honeybees for A and B genotypes of DWV. He said to me at the time, which was 2 months ago, "No, Robert, we only have DWVA in our colonies. We've not screened though for B because we don't think we've seen any change here." These are the data that he sent to me a week ago. Indeed, 2009 to 2013 period, all his honeybees that he looked at, or sorry, I should be cautious. He used next generation sequencing for his bees. All I'm looking at now, what I'm showing you, are the percentage of the reads, the sequences that were associated with DWVA in his honeybees and B. And what you can see is basically there is only A, a tiny proportion of B. For the bees that he's been analysed over 2018 to 20 period, the last couple of years, he's hardly got any A. Most of the reads are B. It looks like he's got a complete takeover in Italy. So rather than there being an equilibrium reached in which both A and B are very common, and I guess that's probably what's going to happen with Omicron and Delta, that they both become very prevalent across the human population. unless vaccination tends to knock one of them out. With these viruses, it really looks as though there's some sort of interaction going on between A and B, which means that one takes over from the other and maybe knocks down the other. And both are virulent, so we shouldn't be complacent. Both are very virulent viruses in honeybees. And we've got the spectre, not of small, analogy of a virus in another animal system would be that of rinderpest, which, when introduced into Africa from India with zebu cattle in the 1880s, spread through Africa and decimated domestic cattle. And I think the analogy with rinderpest actually goes a bit further, because rinderpest, it did not only kill a lot of cattle, it also spread into wild ungulates, and it killed umpteen or unknown numbers of a zebra and gnu or wildebeest. So it wasn't just the domestic cattle, but of course nobody at those times was counting how many wild animals were being killed. Large numbers, it's thought. It's thought that they massively depressed numbers of ungulates, and there's been an association there between drop in the number of ungulates and the drop in the number of Serengeti actually associated. So there's good evidence that that the domestic cattle are transmitting pathogen viruses spilling over into cattle. Okay, I still going? Great. So now I want to deal with the issue of viral spillover, which I said I would get onto in the talk. And of course, the honeybee is not the only bee that exists. Here's a lovely, a beautiful Andrena species from Northern Ireland dunes that visits principally Calluna flowers. So they're also important. They're visiting flowers. They're visiting the same flowers that honeybees visit. So there's been a hypothesis that the honeybee, which is undoubtedly the so-called reservoir population, or the reservoir host for deformed wing virus, there's a hypothesis that virus, which builds up to high titers in honeybees, including honeybees and flowers, may be spilling over into other bee species, like, for example, bumblebees that are visiting the same flowers. Now, the best evidence, I think, comes from our own study in GB, 26 sites where we two species of bumblebee at the same time, visiting the same or in the same area, flowers within the same area. And what we found is that I've shown you this picture on the left before. This is the viral heat map of the viral prevalence in honeybees. And DWV, both A and B, is more prevalent in East Anglia and Cornwall. I've already said that. DWV in bumblebees is at lower prevalence. Only up to 25% of bumblebees are infected instead of 80% in honeybees. But it's in the same locations, suggesting viral spillover. Not only that, if we look at the viral sequences, the sequences of viruses that are in honeybees and in bumblebees at the same site, they are identical. So, for example, take L, which is probably Essex. Here are the sequences in black of the virus that we've taken out of honeybee at that site and that we've taken out of bumblebee. They are identical, but they are different from the virus variants in X in Cornwall, which are identical in honeybees and bumblebees in Cornwall, but slightly different from those in Essex. So the only way you can get this sort of pattern, and this is DWVB, and the same for DWVA, that also seems to be spilling over. The only way to get that pattern is if there's ongoing pathogen transfer. And I call it transfer because we can't tell from these data which direction the virus is going in, but the evidence suggests that it's probably going from honeybees to bumblebees. It's not only deformed wing virus, and I've lost my screen here, I can't see. I think this is the one with DWV here. Also with black queen cell virus, we see the same pattern. In blue is the honeybee prevalence, in red is the bumblebee prevalence of the virus. Black queen cell virus, where it's found in honeybees, it's found in bumblebees, and vice versa. The same with acute bee paralysis virus, slow bee paralysis virus, not so with sac brood virus, which we didn't find very much of. So that pattern with deforming virus is also seen with black queen cell virus, suggesting there is pathogen sharing. Is it spillover? Well, to try and address that a little bit in a little bit more detail, are honeybees here and like this, and bumblebees like this Bombus pascorum here, are they sharing virus at flowers, with the suggestion that it's going from honeybees to bumblebees? Well, I'm going to present to you now some data that we're also writing up. Christine Hesse, she was a master's student in the group. She sampled honeybees and bumblebees at 7 sites around Halle and Leipzig. This is a distance, so they're independent sites. Sampled the honeybees, she sampled bumblebees at the same day, and then she recorded which flowers honeybees and bumblebees were visiting at each site. And using those data, she can set up a so-called bipartite network. It's an ecological network. It shows at the top the bees, honeybee, and 3 bumblebee species, Bombus lapidarius, Bombus terrestris, and Bombus pascorum. And she saw this many, so there's a little bars here. Each of these bars represents, there's 276 bars, and they each represent a single honeybee. And it's connected to a flower that we saw it feeding upon. So Cercium vulgare, for example, here, we saw a couple of honeybees visiting Cercium vulgare. We saw a lot of honeybees visiting Carducus anthecoides. We saw a lot of them visiting Arcticum. These are wild plant species in Halle, where she was at one of her sites. Look at the bumblebees. This Bombus lapidarius shares a lot of flower use with the honeybee. Bombus pascorum shares very little flower use with a honeybee. So we can, with these data, get a measure of niche overlap, resource use, whether it's similar to honeybee or distant from honeybee. And there you can see for the 3 bee species, Lapidarius, terrestris, and pascorum, one is quite similar in resource use to the honeybee, one is less similar, and one is quite dissimilar, Bombus pascorum. Then we can ask, how is viral prevalence? And in this case, I'm going to show you the joint DWV and black queen cell virus prevalence, high in honeybees, and the prevalence of the virus in these 3 bumblebees follows the same pattern of niche overlap, in exactly the same pattern. If I plot those data in a different way, it'll look like this. Here we've got the prevalence of virus in bumblebees on the left, and here we've got the niche overlap with honeybees on the x-axis. The more niche overlap, and this is the line of best fit of those data, the more niche overlap with honeybees, the more the bumblebees have virus in them. Lower niche overlap, less virus within them. So this is again suggesting not only is there pathogen sharing, but it is related to common flower use. If they're using the same flowers, there's a lot of virus being spilt over. And those are data that we're writing up at the moment. Again, another publication I'm writing up. Pathogens. Try and get a handle on this transmission. A PhD student, Anya Tehel, who's working with me, has run a series of experiments where she's experimentally tested for transmission. She has put into cages honeybees that are infected and honeybees that are not infected, or honeybees that are infected and bumblebees that are not infected, or vice versa. Infected bumblebees, clean honeybees, infected bumblebees, clean bumblebees, and look to see whether virus is transmitted. In this case, I'm going to tell you about DWV-A data. DWV-B experiments are underway, as are experiments with black queen cell virus. She also replicated the study, but separated the infected and the clean bees and just allowed a feeding tube to be transmitted between the two to see whether transmission could occur over feeding. The results are more or less the same. The results are these. When a honeybee is infected, it's in red, and it's in a cage with other honeybees, those other honeybees that are clean to start with become infected after 7 days. If the honeybee is infected and it's with clean bumblebees, those bumblebees become infected after 7 days as well. If the bumblebee is infected, we experimentally infect the bumblebee by an injection, and we mix it with clean honeybees, the honeybees don't become infected. The bumblebees that are infected mixed with clean bumblebees, the clean bumblebees don't become infected. So these data suggest transmission happens from honeybees to other bees, including honeybees and bumblebees. It does not seem to happen from bumblebees to honeybees, or if it does, it is at a very low level, and I suspect it's all related to dose. Okay, the big question though is, does that spillover of virus impact the host bumblebee or other wild bee species? And that's a difficult question to answer. Our data using commercial bumblebees has been equivocal in a laboratory setting, suggesting little impact, but we want to extend our studies. We're extending them to look at other bee species and also bumblebees in the field, not in little cages, to see what impact it has. Impression at the moment is that it's a mild impact that it has. I'm going to finish now. I can see I'm running up to the end of my time. Just to say a few words about future and novel control. Let me say a word now here about one control method. I think you've probably heard a lot about breeding for control of tolerance to varroa and tolerance to viruses. I want to mention one novel control strategy that has been devised essentially 10, 15 years ago, but has been recently applied to honeybees, deforming virus and varroa destructor. So I'm changing tack a little bit now to finish. Insects in general, honeybees included, do not have an acquired immune system like us with antibodies. That doesn't exist. Rather, they have a method of degrading viruses and also their own, any double-stranded RNA actually, but they have a method of degrading viruses that enter the cell, the so-called RNAi signaling pathway that leads to the chopping up of virus identical to a primer strand or an initial strand that is given to them as a double strand. So one can feed double-stranded RNA with a specific sequence, for example, a sequence like deformed wing virus, to an insect. Its so-called innate immune system for degrading virus will recognize that sequence, and when it comes across a virus with the same sequence, it will chop it up. So this is termed RNA interference. And this is a method that seems to work very well in the laboratory to knock down gene expression, but also in the laboratory it has been used to knock down DWV in honeybees and also to knock down varroa mites. And just last year, so just over a year ago, this is just the very beginning of 2020, Nancy Moran and her group from the University of Texas in Austin published an article in which they genetically transformed some members of the bacterial community in the gut of the honeybee, the microbiome, to express double-stranded RNA that is identical to deformed wing virus, so that the host, the honeybee, would knock down deformed wing virus. They also designed some to knock down varroa mites, and seemingly the bees became tolerant to varroa mites, and a short-term solution using RNAi that may help over the short term to control virus, including DWV, which is a killer of honeybees, and possibly also varroa mites that transmits that virus before longer-term solutions can be found. So that's some positive note about something that might be possible, feasible to control the major problem, the major scourge of beekeeping. And just coming back to SARS, I mean, thank goodness, SARS-CoV-2, thank goodness we have an acquired immune system, and that vaccination, if it doesn't stop the disease, it certainly reduces the symptoms of the disease and can go a long way towards us getting out of this pandemic that we're in now. I can encourage everyone to get vaccinated. I'm already triple vaccinated as a precaution against SARS-CoV-2, and I think it's advisable to do so. It is our only solution. If only honeybees had an acquired immune system, wouldn't life be easier? So in terms of take-home messages, DWB is now widespread, the major virus associated with varroa mites, and a problem for apiculture because it seems to be a little bit more virulent than DWA, although I should stress both are killers. It seems to be expanding at the expense of A, and it's also spilling over into wild bees. Though exactly what it's causing there for them, what cause it has, what effect it has on the wild bee populations is unclear. I'd like to thank everybody in Halle in the working group that have contributed to these data. There's a lot of people I've not mentioned, collaborators elsewhere, and the funders that you can see here displayed. And I'd like to thank you for your attention. Thank you.
Robert, that was absolutely superb. Thank you very much indeed. It does raise, I think, a fair number of questions. One of the things which I think I would maybe point out to you, in fact, is that you showed a slide of cattle that have died of rinderpest. As far as I understand, I think rinderpest has been eradicated from the world.
You are correct, John. I mean, you should know better than I as a veterinarian. I think it's 10 years ago. One of the UN organisations, Quangos, declared it extinct from the world. That's right, and that's only the second virus that has been that they've done. The first was smallpox. That was about 5, 6, 7, 8 years earlier than that. So we've got one human virus eradicated, except for a few laboratories somewhere in the US or Russia, and rinderpest, which I guess is probably in a few laboratories, high security military laboratories, but eliminated from the world. You're right, John. Yes, and it's possible because they have an acquired immune system and they can be vaccinated against the disease, and precautions can be taken against transmission. Yeah.
Right. Well, you never know. That's one human virus gone and one animal virus gone. Maybe the next one could be a bee virus. You never know. Anyway, that was a fantastic talk, Robert. Thank you very much indeed. It certainly has brought us all up to speed. Because certainly I've been hearing about the deformed wing virus A and B over a number of years, and it all seemed quite confusing. But that has really sort of settled a lot of questions in my mind.
Okay.
Anyway, if we could come on to some questions.
Yeah.
First one here is from David Ferguson. He said, "What was the method used to weigh the bee colonies? Is this something the hobby beekeeper could do?"
Okay, so we measured the size of the colonies using the so-called Lieberfeld method, and I think it was named after, I think it was a Swiss researcher, either his institute or his name. I think maybe it was his institute, which entails taking up every frame and scoring how many bees, how much brood, and how much honey and pollen is on the colony. It's quite a labor-intensive method. More direct would be or simpler would be, and less disruptive to the colony. So that's what we did. We didn't weigh them, we measured how many bees in the colony. Nowadays, there are scales that are available for weighing in real time honeybee hives. We've got 8 of our colonies on so-called beep basis from a Dutch company, and they give us every 10 minutes a recording of the weight, and that gives us an indirect estimate of the size of the colony. I should say we're doing the Lieberfeld method at the same time just to confirm whether the weighing and the size estimates are similar. But the Lieberfeld method is very destructive for the colony. You know, it's very invasive.
Right. Right. So the next question comes from Megan Seymour, who's a prominent bee inspector in England. And she asked, does black queen cell virus affect anything other than queen cells?
Almost certainly it is everywhere. I think most of the time it's being transmitted oral fecally, and it doesn't cause probably much of a problem. It is so it probably reduces the lifespan somewhat of the bees. When we feed it to of adult bees, when we feed it to larvae, where we've done controlled experiments, we find that worker larvae, as well as queen larvae, are susceptible to black queen cell virus. But it's probably a matter of dose. So it is problematic. It probably is killing some larvae if it's at high prevalence and at high titers in adults in a colony. It is surprising how widespread it is. It is almost everywhere, you know.
Yeah, so it would give the impression then that it's asymptomatic in a lot of bees, as you say, maybe getting in some larvae, but it's asymptomatic maybe in the adults. But there could be some losses that we wouldn't be aware of. Yeah,
subtle effects. Yes.
Right. We've got a bit of music for somewhere, right? So from
opportunity to find out.
Oh, sorry. I actually want to stop the questions because we actually have a little video from Simon the Beekeeper.
You can get from it is fantastic. Hi, it's Simon. I'm going to give you an introduction to getting your first beehive. Hi, I'm going to show you how to use Epidemiology. Hi, I'm going to assemble some national frames now. Hi there, I'm going to show you how to use ApiLife Bar, a very widely used, cheap and effective varroa treatment. I'd like to talk to you about getting your bees protected, ready for the winter. So the first thing you've got to be aware of is keeping them warm and dry. Good morning. I'd like to introduce a new addition to our range. It is the pre-assembled foundation and frames. This is the Buzz Professional Bee Suit. It is a result of our designing and manufacturing a premium quality bee suit at a reasonable price. Introducing our new stylish range of Wellington boot, what the up-to-date beekeeper is wearing this year. As you can see, they're bright white. They're not particularly heavy duty, but then you don't want them to be. So £6.99, all sizes, can't go wrong.
Right. So I've cut into that a little bit. I'd like to really thank Simon the Beekeeper very much for his sponsorship. And if anybody is interested, those particular sheep, the breed of sheep that he was keeping there are Valais black-nosed sheep. They're very, very cute looking, I think is the best way to describe them. Anyway, if we could go back then, well, again, to say thank you to Simon. We just may go back to some questions. And it's from Morris, and he said, is there any comparison of virulence of DWVA and B with Asian honeybee isolates of deforming virus?
So not that I know of, no. The variants that they have in Asia are not so different from the variants that we have in Europe. And they are undoubtedly derived from those European variants. Whether there are differences amongst the Asian honeybee species in the viruses that they carry is an open question. And actually, I've got a Thai student with me at the moment, Tanya Lei, that's looking at just that problem.
Right, okay. From Steve, whoops, hold on. Where are they? Yes, does does deforming virus infect queen bees, and if so, can it be transferred to daughters, workers, and daughter queens?
Yes, deforming virus can infect queens, and it has been shown experimentally already in, I think, 2006, 2007, that an infected queen or a queen can become infected by mating with an infected drone, and that an infected queen can pass on deforming virus to her eggs and therefore to the offspring that she lays. So deforming virus has multiple transmission routes, multiple. They are the most efficient, though, is through varroa mite feeding. Right. And that is an extremely efficient, we're talking about 99% efficient form of transmission. Right. Interestingly, deforming virus B seems to be able to replicate in varroa mites. DWVA, genotype A, doesn't. It's merely physically transmitted, not biologically. How much difference that makes, I'm not sure, because DWVA, is already, let's say, 99% efficient at transmission. So B might be even more efficient, but might be injected in by the varroa mite at a higher dose. So that is a paper that came out last year, 2021, Gizda and Genush, showing quite clearly B replicating in the mite.
Right, okay. Because I'd heard something that say is that there was consideration maybe is that the viruses may be able to replicate without the use of the varroa mites, and they could still replicate in huge quantities. I don't know if that is true or not.
But it's, normally probably deforming virus is transmitted fecally orally. So if we inoculate by feeding, we cause an infection of deforming virus in a honeybee, and it doesn't live so long as a honeybee that's fed with just bee juice. So therefore it does cause mortality, but only slight. The problem comes when it gets into the hemocoel. This is a problem for insects. They have a single cavity, the hemocoel, where all the blood, the hemolymph flows through, that's all organs are in contact with. And as soon as some pathogen gets into that, it's chaos inside the bee. There's a parallel there with polio. Polio in humans is fecal oral, normally transmission. If it gets into the nervous system, then it causes laming. It causes polio disease, and with terrible consequences, possibly also death.
Right. This from Steve. What are the mechanisms that allow honeybees to survive these viruses, or are more tolerant to them?
So we don't know for certain. The viruses induce an immune response. We find that DWVA induces a response. DWVB seems not to, seems to be silent, which might explain why it's partly able to replicate so well. But I am convinced that there are differences in the tolerance of bees, in their susceptibility to viruses, that some may be able to suppress virus or may be able to withstand higher titers of virus than others. So I'm sure there's genetic variance there amongst honeybees in their ability to fight the virus or to tolerate the virus. And I think that has been suggested also by some experiments that have shown that honeybees survive long periods of time with varroa mites. For example, the bond bees from Gotland. Actually, they don't seem to show any different. The varroas on them don't seem to show any difference. They don't seem to show any difference to varroa. But they seem to be a little more tolerant of virus, or somewhat refractory to virus compared with non-susceptible honeybees. So, in fact, when people tell me that they've got honeybees that can survive varroa, I suspect what they've got is honeybees that can survive virus more than varroa. But anyway, that's another kettle of fish that we can get into if we.
Yeah, well, what I would think, sure, that would be quite something, because you say if we're getting pockets of honeybees that are tolerant to varroa mites, could they then become tolerant to the viruses? Obviously, so yeah, that could be for the future. Anyway, Samantha asks, so could these diseases be passed more to bumblebees through commercial breeding of bumblebees for pollination purposes, as these bumblebees are fed pollen harvested from honeybee colonies?
Yeah, not only are they fed honeybee pollen collected from honeybees, at least originally, the companies that were rearing bumblebees initiated, it's difficult to get a queen bumblebee to start a nest. And the one trick they used is to put 3 or 4 worker honeybees, nurse bees, in with the bumblebee. So immediately you've got the possibility of transmission. We purchase commercial bumblebees for our experiments. We have to screen them for virus. So we find black queen cell virus, deformed wing virus, maybe sac-brood virus in them sometimes. And then we have to eliminate that colony and not use it in our experiments. So yes, bumblebee colonies, commercial colonies, certainly could be a source of pathogens. And not only that, it's already been shown 2014, 2015, Peter Greystock et al., that bumblebee colonies, commercial colonies, sometimes contain nosema disease. And I think they've also found foul brood, if I recall correctly, in those bumblebee colonies. So actually they could be carrying, even if they're not susceptible to those diseases, they could indeed be transporting disease of honeybees. And that's why there's been a call for greater control of diseases in those bumblebee colonies. I think the companies respond to that. I don't know exactly how they do the screening, but they are certainly aware of that issue, a potential issue, in that the pathogens in that case, if it's AFB spores, for example, they of course could spill back into honeybees.
All right. Is there any evidence that bees can develop herd immunity, just as in the human population? It's very relevant to COVID as again.
So an interesting idea. I think when it comes to social insects, one would talk about social immunity, and many social insects have a number of traits that they exhibit which give them immunity. For example, some ant species use their venom gland, formic acid and other compounds, to protect themselves against fungi. And that's been well demonstrated that they use that. So and they spray that on their neighbors as well, their nestmate sisters, to protect them against fungi. The same is thought for termites. With honeybees, they do have some forms of social immunity. For example, when defending themselves against wasps or hornets, they can ball a hornet, at least some species of honeybee in Southeast Asia, and also maybe some subspecies of honeybee, Apis mellifera, can do the same. They can, it's been suggested that they can warm the colony up as an act of fever, if you like, and they use propolis extensively, and they groom one another. Whether these factors can help against virus is not clear. Maybe indirectly in terms of reducing varroa numbers, because that's the easiest way nowadays to stop virus or to reduce virus. Kill varroa mites, get rid of varroa mites, because they are the main problem with these viruses, that they're now being transmitted by the varroa mites. Yeah.
Right, okay. This is from Gavin Ramsey. He said, in the absence of good data, do you have any hunches on whether viruses spreading from honeybees to wild bees do cause real problems? And he's trusting your hunches here.
Yeah, okay. So people often report bumblebees with deformed wings. And the first report of DWV in a bumblebee was from Elke Genusch in 2005. She picked up bumblebees with deformed wings in a honeybee apiary. So in an apiary. Exactly how they got the deformed wings is not clear, but they were certainly packed full of deformed wing virus. and they were associated with the honeybees in some way, in that they were living nearby. So that's indirect evidence. But I know that in Germany, there's even a website where people who report bumblebees with deformed wings can sign in and monitor their deformed wing bumblebees. And these are apparently quite prevalent, a trait. Whether all of those are caused by deforming virus is not clear. So my hunch is that it has mild effects, yes. How serious, I can't tell. Our experiments in laboratory show no effect, or only subtle effect when the bumblebee is stressed, when we remove food and they're short of food. So the question is what's happening out in the field, because bumblebees in the field are much more stressed than in our little cages. In our little cages, they have a surfeit of sugar syrup and friends to play with or to interact with. Out in the field, they're constantly flying under pressure to collect nectar and pollen for the colony. So my hunch is mild effect, and probably variable across across bee species as well, I suspect.
Right. Okay. I think we will make this the last question. It's from Andrew Shinnick, but it's really just to say is that I'm sorry we just don't have time to go on with the questions, and we could probably go on all night. But certainly, if you have any real questions that you would like to ask Robert, I think we'll move it up something on the screen with your email address on it, Robert, and people can contact you directly, which we are much appreciated of.
Yes.
Okay. The very last question here is from Andrew Shinnick, and he says, "What are your thoughts on treatment-free beekeeping? Should we allow bees to figure it out, or should we scientists intervene in order to prevent other pollinators from getting these viruses?"
That is going to have a very long answer. So I think we're going to have get out a couple of glasses, John, and sit and talk about that one, because there are pros and cons. You know, I think ideally it would be great to have treatment-free beekeeping. As I recall, when I started back in 1985 in Cardiff, you know, what glorious days they were on reflection, with the only problem being occasional nosema. The problem at the moment is I fear that if people were to completely stop treating, we might lose 99.9% of honeybees. Yeah. And, you know, that's my fear. Now, I've heard in Ireland, there are wild colonies living in the wild that apparently have existed for 3 or 4 years. I've seen recently reports from the University of Galway, research being undertaken, suggesting there are pockets of honeybees that seem to survive without treatment. Tom Seeley is well known in upstate New York for having a population in a forest, Arno Forest, where apparently they've been surviving, with honeybees surviving for several years. In Germany, I hear reports repeatedly nowadays of people telling me they have populations that survive. I've heard the same in GB. I don't know, you know, I haven't seen really good data to support that idea. My fear is that treatment-free at the moment would be too much of a hard thing. We would probably lose varroa mites, or at least have them severely reduced, simply because population density of honeybees would be low. And theory predicts that if the population density of a host is low, the prevalence will be extremely low of the parasite as well. And if we lose 99.9% of honeybees, most of those that survive, there'll be odd pockets that survive have very little virus. So or varroa, or maybe no varroa associated with them, but there may be re-establishment. So that is a difficult question to answer. Ideally, yes. I think in the short term, too extreme. Yeah. At least in temperate climates.
Yeah. I think that's probably an ongoing project, because I think it's Professor Grace McCormick at the University of Galway who's asking for samples from bees, or are not treated at all, or beekeepers who are doing it. And as I say, I think she is getting quite a response, but that's an ongoing thing. We won't know the answer to that for quite some time.
Yeah. Anyway. No, Thank you, John, for naming Grace. She's got an interesting research program underway. Yes, very much so.
Anyway, I come down to Robert to thank you so much for what I thought was a wonderful presentation, and to everybody who, I'm sure you will have, they will all appreciate that was an excellent presentation. So, and really say, and again, really just to thank you so much for this. And it's really just for me to say is that next week, we have Professor Jamie Ellis from Florida, and he will be talking about the swarm reproduction at the colony level. So if you wish to tune into that, then we're going to see you here at this time next week. And then on behalf of UBKA, I'd like to thank you all for listening. And again, thanks to Robert Paxton, and we'll bid you good night.
Thank you, John. Thank you for moderating so well for me. Thank you.
Well, and
the introduction.
Okay.